वेलकम टू द लाइफ साइंस लर्नर्स। तो कैसे हो दोस्तों। आज हम देखेंगे डीबीटी का सिलेबस ऑफ द डीबीटी जेआरएफ एग्ज़ाम। मिलते हैं इंट्रोडक्शन के बाद। दोस्तों, सिलेबस फॉर डीबीटी जेआरएफ एग्ज़ामिनेशन इनमें दो पार्ट होते हैं, पार्ट ए एंड पार्ट बी। पार्ट ए में आता है जनरल एप्टीट्यूड एंड जनरल बायोटेक्नोलॉजी, एंड पार्ट बी में आता है जनरल प्लस स्पेशलाइज्ड ब्रांच बायोटेक्नोलॉजी। पार्ट ए में 15 कंपलसरी क्वेश्चन होते हैं जिसमें आपको 15 सॉल्व करने होते हैं, एंड देन पार्ट बी में आपको 150 क्वेश्चन गिवन होते हैं जिसमें से आपको 15 क्वेश्चन कंपलसरी सॉल्व करने होते हैं। पार्ट ए इंक्लूडेड कंपैरिजन बेस्ड क्वेश्चन जिनमें आते हैं विच इंक्लूडेड्स पैराग्राफ, देन टेस्ट नॉन वर्बल रीजनिंग कैपेसिटी जिसमें इंक्लूडिंग फाइंडिंग द ऑड वन आउट इन अ सीरीज ऑफ ऑब्स्ट्रैक्ट पिक्चर्स, देन जनरल बायोटेक्नोलॉजी इसमें आते हैं इंक्लूडेड्स बायोमोलिकुलर स्ट्रक्चर एंड फंक्शन इंक्लूडिंग covalent structure, then force that stabilize biomolecules, then protein structures, conformation of nucleic acid, then thermodynamics, then physical properties of water and their role in biology, then chemical kinetics, then enzymology, then methods in biotechnology which includes concepts of precising and accuracy in experimental measurements, then biostatistics, then biochemical methods, then UV visible spectrometry, then fluorescence spectroscopy, then fundamental CSIR and Raman spectroscopy and their use in the study of biomolecular conformation, then centrifugation, then microscopy, and then fundamental of X-ray, NMRA, nuclear magnetic resonance and cryo-electron microscopy for determination of biomolecular structure. Then organization of structure and functions of prokaryotic and eukaryotic cells, which includes cell wall and cell membrane. Then structural organization and functions of cell organelles, then organization of genomes, then cell division and cell cycle, then cell transformation and cancer oncogenes and proto-oncogenes, tumor suppressor gene metastasis, then cellular processes which includes DNA replication, repair and recombination, then transcription of various type of RNAs and their processing and modifications, then protein synthesis, processing and transport of proteins, then control of gene expression at transcriptional and translation level, then host pathogen interactions, then cell signaling, then cellular communication, then innate and adaptive immune systems, that is immunology, then recombinant DNA technology which includes enzymes used in recombinant DNA technology, then isolation and purification of DNA and RNA, then biomolecular cloning of DNA or RNA fragments in bacterial and eukaryotic systems, then generation of genomic and cDNA libraries, then isolation and amplification of specific nucleic acid sequences, then DNA sequencing methods, then methods for analysis of gene expression at RNA and protein level, then analysis of DNA polymorphism, then biosafety regulation and IPR, then genetics, phylogeny and evolution, which includes chromosomal inheritance, then extra chromosomal inheritance, gene concepts, genetic analysis, mutations, DNA fingerprinting and applications, DNA barcoding, marker assisted selection and QTR mapping, species concept in archaeal, bacterial and eukarya, phylogenetics analysis and evolutionary relationship among taxa MLST, then genomics and proteomics, which includes introduction of genomics, then genome sequencing, then DNA microarray technology, then proteomics, then IPR, biosafety and bioethics, in which types of IP, then invention in context of prior art, then types of parents, Indian Patent Act 1970, then patent applications, then biosafety, then bioethics. तो यहाँ से होता है पार्ट बी शुरू. पार्ट बी इंक्लूडेट्स स्पेशलाइज्ड branches in biotechnology, which includes agricultural biotechnology. In this, plant biology, physiology, molecular biology and seed technology, which includes plant cell structure and function, then hormonal control of seed germination and seedling growth, then photosynthesis, then nitrogen fixation and regulation, role of lectins, then biosynthesis of secondary metabolites, then light control of plant development, then solute transport and photoassimilate translocation, then plant microbe interaction, then seed science and technology, then tissue culture, transgenic technologies and biotechnology, which includes totipotency, then production of alkaloid and other secondary metabolites, then direct transformation of protoplast using PEG, then promoter, plant selective marker, reporter gene, then genetic engineering of crops, then biofuels, then molecular breeding and genomics, which includes principle of plant breeding for self and cross pollination pollinated crop, heterosis breeding, limitations of conventional breeding, molecular marker, then gene flow in plants, then development of ETS, ESTS, then marker assisted breeding for various traits, then genome organization, then abiotic and biotic stress biology, abiotic stress, then biotic stress, then biodiversity Act 2002, then biodiversity legislation in India. Then impact of GE crop on biodiversity. Then animal biotechnology, which includes animal physiology and biochemistry. Then histology and embryology. Then animal parasitology. Then animal virology. Then immunology. Then pathology. Then animal genetics. Then molecular medicine and surgery. Then molecular diagnostics. Then research methodology. MTech biotechnology and biochemical engineering and industrial biotechnology, which included subjects are industry mathematics, engineering principles, thermodynamics in biological systems, then bioprocesses engineering and technology, then downstream processing in biotechnology, then bioprocess in plant design, then bioinformatics and computational biology, which includes major bioinformatics resources, database research, then sequence analysis basic concepts, then scoring matrix, pairwise sequencing, alignment, multiple sequence alignment, application in taxonomy and phylogeny, comparative genomics, then structural biology, then proteins, then DNA and RNA secondary and tertiary structure, tRNA tertiary structure, then classification and comparison of protein 3D structure, then application in drug design, then environmental biotechnology, which includes basic ecological concept and principles, then chemistry of organic and inorganic chemicals polluting environment, then environmental pollutions, then water pollution, then wastewater treatment system, then treatment of typical industrial effluent, then management of municipal, biomedical and agricultural solid waste, then environmental pollution control, then environmental friendly technology, then pollution monitoring, then global environmental problems, then marine biotechnology, which includes important marine organisms and their behavior, then marine resource assessment, then population study and marine environment protection, oceanography, then types of marine microbes and their biology, then microbial assessment, then role of microbes in marine environments, then microbial metabolites, then microbial interaction, then culture system and hierarchical techniques, then introduction to marine pharmacology, then manipulation and microbial techniques, then disease diagnosis, then marine organisms and environmental interactions, then population and biomaterial interaction, then fouling and corrosion, then wastewater biotreatment, then medical biotechnology, in which vaccinology, then immunotechnology, then CMI and imaging technology, genetics in medical practice, then genetics of neurogenetics disorders, then genetics basic of eye disorder, then complex polymorphic syndrome, then component of genetic counseling, then infection of the gastrointestinal tract, then infection of respiratory system, then pharyngeal larynx illness, pharyngeal illness, then infection of nervous systems, then sexually transmitted disease and congenital infections, then host pathology interaction in disease processes, then DNA based diagnosis, then antibody based diagnosis, proteomics based diagnosis, then gene therapy, then cellular therapy, then recombinant therapy, then immunotherapy, then gene silencing technology, then human disease genes, the molecular and human genetics, which includes cell biology and cryogenetics, then principles of genetics, then common genetic disorders, then human genome, the neuroscience, in which including neurons, glial cell, neurophysiology and behavior, then major event in early embryonic development, then patterning, polarity and reorganization of nervous systems, then birth and migration of neuron, then neuronal death during development, then neurotropic factor, then electrical properties of excitable membrane, then electrical properties of neuron, quantitative model of stimulation, Hodgkin and Huxley analysis of Scott giant axon, then synaptic transmembrane and cellular signaling, then sensation and precipitation, then fundamental of motor system, then chemical and control of brain and behavior, then neural control of breathing, then neural control of heart, cardiovascular homeostasis, then sleep and dreaming, cognitive development and aging, clinical neurochemistry and neuropathology, then pharmaceuticals biotechnology, which includes drug metabolism, then pharmacology screening and assay, then enzymes and microbial technology, then biochemical engineering, then downstream of processing, then industrial applications. तो दोस्तों सिलेबस का वीडियो थोड़ा बड़ा हो गया है। अगर आपको सिलेबस का स्क्रीनशॉट चाहिए होगा तो आप ले सकते हो वीडियो पॉज करके, अदरवाइज़ मैं इसकी पीडीएफ की लिंक आपको डिस्क्रिप्शन में दे दूँ। आपको इसकी पीडीएफ की लिंक डिस्क्रिप्शन में मिल जाएंगी, आप वहाँ पे जाके पीडीएफ डाउनलोड कर सकते हैं। थैंक यू। थैंक यू फॉर वॉचिंग दोस्तों।